Good morning, everyone. So, in today's class, I'll continue my biomolecules chapter, and in today's video, I'll talk about the nucleic acids. So, nucleic acid is very known thing to all because we all have heard about DNA and RNA. So, yes, in today's class, we'll talk about DNA and RNA as nucleic acids, and we'll also talk about how they were discovered and their structure, their functions, everything. Okay, so let's start. First of all, about nucleic acids. So, nucleic acids, just like other biomolecules, it is also a biomolecule and it is actually a macromolecule. Okay, so nucleic acid is a long chain. You can talk about DNA or RNA, whatever. So nucleic acid is a long chain, and in this long chain, many monomers or repeating units of nucleotides are forming a long chain of polymers. Okay, remember that. And also, this nucleic acid is very important in cells and viruses because they carry genetic information which is required for making RNA and proteins, and which is required for the functioning of a living thing. Okay, now nucleic acids are of two types, DNA and RNA. So first we'll start with DNA. So DNA history. See, DNA in the year of 1869, a person known as Johannes Frederick Mischer, what he did, he identified DNA, okay, but he named it as nuclein. So he saw this DNA in a nucleus of a cell or WBC, okay, white blood cells. He named it as nuclein. That time the name was not yet given as uh, DNA, okay. So then Altman, he changed his name into nucleic acid, okay. Then another person known as Kossel, he saw that nucleic acids, they contain purine and pyrimidine bases, sugar and phosphate. Then next, very important part in this history of DNA is the discovery of the structure of the double helical structure of DNA. Okay, it was done by James Watson and and Francis Crick. Okay, in 1953. So they discovered this double helical structure. Also, Kossel, as I've told you, he said that nucleic acid contain purine and pyrimidine bases and sugar and phosphate. He also got the Nobel Prize. Okay, he got the Nobel Prize because he named this nuclein as DNA. Okay, he named nuclein as nuclein. He named this nuclein as DNA, and he also isolated the five nitrogen bases that we know as adenine, thymine, cytosine, guanine, and uracil. So he have isolated these five nitrogen bases from DNA. Okay. Now, next we'll talk about RNA. RNA was discovered before DNA, okay? Because when this discovery of nuclein was going on, similar in that same time, also this RNA was discovered, okay? Therefore, RNA is also the first genetic material to be discovered. And RNA's role as in protein synthesis, we know that RNA is useful for protein synthesis, okay? So that role was discovered in 1939, and Leslie Orgel, he is known as the father of RNA because he has worked with RNA. He have worked he has told many things about RNA. Okay, so basically he is the father of RNA. Remember that. And Dr. James D. Watson, he is the father of DNA science. So this is all about DNA and RNA, which you should know. Moving on to structure of DNA. Now, if you can see the structure out here, this is double helical structure. Okay, because uh, you can see here the structure of DNA. Okay, when we look, we will see DNA is like this. It's folded. Okay, so it's in helical structure. It's folded. Deeply, I'll tell you in my next slide. So now, what is happening in DNA double helix? See, I'm just giving you a basic structure out here. So if we break it a bit, we will know that inside this all are the base pairings. Okay, these are the base pairings, colorful. So moving, come to the previous picture out here. See, this colored ones are pairing. Okay, base pairing. This is ATCG in DNA, ATCG. Okay, but in RNA it is AUCG. Okay, so out here A and T, adenine and thymine, they are forming bond. Okay, double bond C, two bonds. Then C and G, cytosine and guanine, they are forming. Guanine is forming three bonds. Okay, see three pairs. Okay, out here. Like that only it will continue. Okay, it will continue. ATCG will continue till the end. It will this base pairing is forming this helical structure. Okay. Now, if we break it further, we will know that, as I told you about nucleotide. What is nucleotide? Nucleotide is a structure, or you can say it's a composition of phosphate group, sugar group, and the bases, four nitrogen bases. Okay. In case of DNA, it's ATCG, and in RNA, it's AUCG. So this is the nucleotide. Nucleotide is basically the phosphate, sugar, and base. Remember that. So I hope you have understood till here. So this A out here, this is A out here. See, looking in the previous one, this sugar, this is base, okay? This base is linked with the sugar out here. Here there's a sugar out here, okay? Then the sugar is linked with the phosphate out here. Structure goes like this, okay? In both sides, it go like this only. So this way, the entire structure of DNA is formed. Now we will know more details of this helical structure. Now we'll talk about double helical structure in details that was given by Watson and Crick. Okay. Now if you see here, see the structure out here. See, this is the base pairing. I told you in my previous slide, base pairing. Okay. The DNA is running in the you can say out of from 5 dash to 3 dash in this side, and this is the 3 dash to it is anti-parallel. Okay. So both are parallel, but but the direction is opposite in both sides. 5 dash to 3 dash and 3 dash to 5 dash. Okay, that way. Then this is the you can see the inside of the DNA. The base pairing is there. GC80 base pairing is there. Now this nitrogen bases, they are having there are two groups. Okay, major group and minor group. Okay, so in major group and minor group, you can see out here between the adjacent base pairs. Okay, like suppose between this and this, the distance is how much? 0.34 nanometer. Okay, the distance is 0.34 nanometer. But in each helical turn, there are 10 such 10 pairings. So for the 10, it will be 3.4 nanometer. Now, the diameter of the DNA. Diameter of the DNA is 2 nanometer. Remember this, very, very important. 0.34 nanometer between two base pairings, and the diameter is 2 nanometer. This is structure of DNA. Fine. Now come to the right side picture. You can see here, I've already explained it in my previous slide, but once more I'll do it for you. See, here it is 80 group. Okay, this is the pairing between two nitrogen bases. Then you can see on both side is a sugar. Okay, so this T or A, this whatever the base is there, nitrogenous base is there. This this base is linked to the sugar in the first carbon. See the diagram of sugar is like this. Okay, so there are 5 carbons. Okay, there are 5 carbons. So in the first carbon, it's 1, 2, 3, 4, 5. The first carbon out here is linked with the nitrogenous base. Okay, and the fifth carbon. Okay, the fifth carbon is linked here with the phosphate group. Okay, so out here the phosphate group is linked with the fifth carbon, and the base is linked with the first carbon. So the first carbon link is known as glycosidic bond, and since the phosphate is acidic in nature, therefore the structure is known as nucleic acid. Remember that. I hope this much is clear for you. Now we'll talk about the link or the bond that is formed out here, known as phosphodiester bond. Okay, what is phosphodiester bond? When the phosphate residues out here, you can see a phosphate group. When they form a link between the 3-hydroxyl group of 1-deoxyribose. Deoxyribose is what? A sugar. Okay. So when there is a bond formed with the 3-hydroxyl group of 1-deoxyribose with the 5-hydroxyl group of the next one, then it's known as this one. It is known as phosphodiester bond. Okay. So there's a phosphodiester bond. See 3 dash 5 dash. Okay. See 1, 2, 3. So 3 dash, and this is the fifth one. Okay, so fifth one is this one. So third and fifth, when there is a linkage formed, or then it's known as phosphodiester bond. So remember, this is the structure of double helix. Okay, of DNA structure. I hope you have understood this very, very clearly. Moving on, Chargaff's rule, very, very important. Erwin Chargaff, he gave a rule where by which you can understand the structure of the double helical structure of DNA. Okay, you can understand the structure of DNA. So he said that the amount of one base is equal to the amount of the second base. That means we know that there's a pairing between purine and pyrimidine. Okay, purine is out here, purine and pyrimidine. So he told that the amount of purine will be, the purine base will be equal to the amount of the pyrimidine base. Okay, understood this one? So in a double stranded DNA, that means the amount of guanine is equal to the cytosine base. Okay, so understood this one? I hope so. So that means adenine and thymine, they form a bond A and T, and C and G form bond. So amount of one base A will be equal to T. That's why they're forming a bond. And similar way, amount of C will be equal to amount of. I hope you have understood that. Okay. So this composition of thing, they will. Okay, now there are different species, right? So for different species, this composition of DNA will not be same. It will vary from species to one species to another species. Okay, have you understood this? This is all clear, I hope. Okay, it's all clear. Moving on now. So adenine and guanine, okay, they are the purine base, you know that, and cytosine and thymine, they are the pyrimidine base. So remember that also. Now moving on to forms of DNA. Now there are five forms of DNA: A, B, C, D, and Z. Okay, so B DNA, B type of DNA is the most stable and the most active DNA. So most usually generally our DNA is the B DNA type. Okay, because the calling direction is right. Remember that the base pair is 10 base pairs. I've already told you there are 10 base pairs in every turn. Okay, then also the sugar phosphate backbone is smooth. The pitch of helical 34 angstrom, diameter is 20 angstrom, stability, I've told you it's stable and active. Remember this if there's a question coming to you, which is the left, in which type of DNA this left coiling or left helical, you can left helical structure type, okay? Then the answer is Z DNA. And if they ask you more stable and active DNA, then it is the B DNA, okay? They might ask you which DNA has 12 base pairs per turn, then you have to say it's a Z DNA, okay? Or which DNA has a zigzag backbone, to know that ZDNA. Okay, so these are some questions that may come from this types of or forms of DNA. Moving on to functions of DNA. In functions, I've already told you DNA is a genetic material. You can see in my other classes of DNA and RNA. Also, I've taken another class of DNA as genetic material. You can see that class, very helpful. Okay, so DNA is a genetic material. It will pass information from generation to generation. As we know, when a child is born, a child inherits some characteristics such as just like the father or the mother. That means it is inheriting characteristics from his parents. So what is this characteristic? Basically, it is the genetic material. It's the genetic information that is being passed from a parent to an offspring through the DNA. Okay, so DNA will keep the information intact the next generation. That is how we say now that you have her genes, you have her genes, your father's genes, you have your mother's genes, because this gene in genetic information is being passed and it is intact. Okay, next, now it also helps in mutation. So you know DNA undergoes mutation, and and mutation can lead to many other things. Okay, so also DNA, it's like helping in cellular metabolism, and also very important translation of essential protein for proper functioning. So you will know about that. DNA is undergoing DNA is being transferred to mRNA for transcription. So there are many processes occurring. So DNA has a lot of many functions. Main function being the genetic information. Okay. Moving on to RNA. So as I've told you in my first slide that RNA is the first genetic material that has been discovered. Then RNA is present in a majority of living organisms and viruses. See, before when DNA was not yet discovered, still they were living organisms, right? There were viruses were discovered very earlier. Then there was also bacteria was there, archaebacteria was there. So that time DNA was not discovered. So there was some genetic material. So that time it is said that RNA was present, and RNA was the first genetic material. So normally, like basically, RNA is mostly it exists in the form of single-stranded, but DNA is double-stranded. Okay, but in RNA is mostly it is single-stranded. But there are some RNA viruses which are also double-stranded in nature. They're special RNA viruses. Okay, now in this RNA there are A, G, U, C. Remember, I told you thymine is replaced by uracil. Okay, so in RNA we have A, adenine, then we have uracil, and then we have cytosine, and we have guanine. Okay, these four are the nitrogenous bases present in RNA. Now, even here, same like before, it is composed of nucleotides, and nucleotides are attached to sugar. Okay, nucleotides are out here, same like before. The nucleotides are composed of sugar is there. Okay, then phosphate groups are there, and also nitrogenous bases are there. Okay, remember that. Now, RNA stands for ribonucleic acid, you know that, and DNA stands for deoxyribonucleic. Okay. The types of RNA. Now, this basically there are three types of RNA: mRNA, tRNA, RNA. Now, Francois Jacob and Jacques Monod. Okay, they discovered this mRNA. They have given the mRNA. So mRNA stands for messenger RNA, mRNA. Okay, the M you can see out here. The M stands for mRNA. See, this M stands for mRNA. Okay, mRNA is, sorry, messenger RNA. Okay, M stands for messenger. T outer stands for transfer RNA. R stands for ribosomal RNA. Okay, So mRNA, tRNA, rRNA. These are three types. Now, messenger RNA, what does it do? If we see the structure of the, see, I've given a structure out here. Just look at the structure out here. Okay, see this M out here. This is the mRNA. Okay, this is the mRNA. So this is the messenger RNA. It contains some messages. Okay, remember that. This lower part, you can see this one. This is the rRNA. Okay, it's ribosomal RNA. And this one, yellow one, you can see that is the transfer RNA, tRNA. Just look at the picture once, keep it in your mind. Okay, then we'll go back to the previous slide and then I'll tell you what it is. So just the tRNA, it is a physical link between mRNA and protein. So see, this is the mRNA and protein is being formed. This is the protein. See out here, not this one, sorry. Here the protein will form this side. Okay, protein will form this side. It's not shown in this diagram. So it is a link between the mRNA and protein because tRNA they will bring the codons. Okay, they will bring the codons which will be identified, which will come and join with the mRNA, and it will lead to the translation process. Okay, so mRNA will act as the carrier of genetic information. It will carry the genetic information mRNA. Okay, and tRNA will is the physical link. It will bring the codons that needs to come and bind with the mRNA. I'll talk about this in my other class of transcription. Here, just remember this much. And our RNA is present in ribosome because this ribosome, see, it will hold the mRNA and the tRNA. The entire structure is being held by this rRNA. Okay, now, so RNA helps in protein. So by this much you've understood that RNA is helping in protein synthesis because this entire process, okay, entire process, what is this entire process? The transcription process. Okay, translation process. Entire process is the translation process, and translation means where protein is formed. Okay, so RNA is helping in protein synthesis. mRNA is providing the template or the genetic information. Okay, tRNA, it is bringing in amino acids. See out here, these are the amino acids: TRP, tryptophan, lysine. Okay, so these are all brought by this tRNA. It's by bringing those amino acids and binding it with the amino acids present in this messenger RNA. Now there is again some techniques where it has to identify the start codons. Many things are there out there: start codons, stop codons. We'll talk about it in my translation. That's not here. Okay, just remember that tRNA is reading the genetic code out here in messenger RNA and helping in the translation process. Okay, rRNA out here, it is a structural role. It is a structural role or a catalytic role. It is holding the entire structure. It is helping in the functioning of the, not functioning, it's helping in the performing of the process. Okay, process occurring, it is holding it. Okay, so this is about your functions of RNA. Now we'll talk about DNA and RNA. So in this slide, we'll talk about the difference in structure. Okay, how is DNA structure different from RNA structure? So as I've already told you, see, just imagine if the bases are different. So that is again forming a structure, right? So in DNA, as I've told you, that it's containing thymine. Sorry, it's containing thymine, and in the RNA, it's containing uracil. So this is the first difference. Okay, here, the thymine and uracil, first difference. Okay, remember that. So now out here is forming a helical structure. Okay, helical structure is formed by sugar and phosphates. They are also same, but here it is single stranded. See, this single stranded, and here this is the double stranded. So this is the difference out here: single stranded, double stranded. Also, the uh, the base that is present out here is different. Okay, here bonding is between A and T. Okay, base pairing is A and T out here. Here, what is the base pairing? A and U. Okay, not T. So that is a difference out here. Okay, remember that. And yet there's a double helical structure. This is the DS double stranded. This is single stranded. Remember that. Okay, this is the difference out here. Now, if there's a question comes out here, DNA or RNA, which is the better genetic material or which is more stronger? The genetic material which is better, we cannot say that because both are containing genetic information and they're present in different organisms. But DNA and RNA among them, which is more stronger? So DNA is more stronger among these two because first of all, RNA is very labile. It's more labile than DNA actually, and it can be easily broken. Okay, so and even when it's broken, for repairing process, DNA is more easily repaired than DNA, so than RNA. Sorry. Okay, so RNA is easily broken, and also the repairing is very not easily repaired compared to DNA. So DNA is not easily broken and DNA can be repaired very easily. So this is very, this is also One of the reason why it's more stronger than RNA. Also, DNA information is very, very protected. Okay, and also since DNA has double strands, RNA doesn't have double strands. So DNA has double strands. Therefore, double checking. It is allowing in double checking. Double it's checked twice since it's containing double strand. Therefore, RNA is much more stronger than sorry, DNA is much more stronger than RNA. Okay, so DNA is easily repaired. It is not easily broken. Okay, it contains double bond, which is allowing double checking, and the information is DNA is more protected. Therefore, DNA is more stronger than RNA. Fine. Moving on to facts on nucleic acids. So, see, this are some of the facts which this is GK facts, or it can come for exam also. What is the E. coli size of DNA? Now, DNA in our in humans or in living things, DNA is present in first. It is, you know that we have humans have 20 pairs of chromosomes, right? So their DNA is present. Also, DNA is present in the mitochondria. Okay, mitochondria also contains DNA. We have seen that. So therefore, now we'll talk about the size. So E. coli, this is the bacteria. E. coli size in E. coli, the size of DNA is 4.6 into 10 to the power 6 base pairs. Okay, size. And in nucleus, nuclear chromosome size, it is ranging from 50 to 250 into 10 to the power 6 base pairs. And in mitochondrial DNA, it is 17 into 10 to the power 3 base pairs. Okay, so this is all about your size of nuclear chromosomes, mitochondrial DNA, and E. coli size. So I will end the video out here. So if you have liked today's class, click on the like icon and do subscribe to my channel for more such videos. Thank you and have a very nice day.